The History and Future of Taxonomy, part of the Systematics and Biodiversity course offered by Leiden University in 2020. I'm Jeremy Miller. In this lecture, we will review the history of human efforts to catalog the diversity of life on Earth and how the advent of the World Wide Web has made biodiversity knowledge more accessible and powerful. People have always had a need to recognize the species they share their environment with, which species are dangerous, which are edible, medicinal, useful. When we speak about the history of taxonomy, we usually mean the history of the Western world, starting with the Greeks. However, the earliest traces of what might be seen as taxonomic scholarship are not from the West, but from the East. In the Eastern world, one of the earliest pharmacopoeias was written by Shen Nung, emperor of China around 3000 BC. He was a legendary emperor known as the father of Chinese medicine. He wanted to educate his people in agriculture and medicine and is said to have tasted hundreds of herbs to test their medicinal value. Eastern taxonomic works were not known in the Western world until the Middle Ages and could thus not have influenced the progress of Western sciences. In the West, the Greek philosopher Aristotle was the first to classify all living things, and some of his groups are still used today, like the vertebrates and invertebrates, which he called animals with blood and without blood. He further divided animals with blood into live-bearing and egg-bearing, and formed groups within animals without blood that we recognize today, such as insects, crustaceans, and mollusks. In the Renaissance painting by Raffaello Sanzio entitled The School of Athens, Plato on the left gestures to the heavens, representing his belief in the forms, which could be discovered through reason. Aristotle gestures to the earth, representing his belief in knowledge through empirical observation and experience. It was not until the end of the 16th century that taxonomic works became original enough to replace the ancient Greek works. One of the reasons for this was the development of optic lenses, which made it possible to study details of different species. Collection of specimens became part of the growing sciences, and emphasis turned from medical aspects to taxonomic ones. This coincided with the age of exploration, which saw Europeans all over the world engaging in commerce, colonialization, and conquest. Samples of the many kinds of unfamiliar things they encountered were shipped back to Europe, including collections of plants and animals. Carl Linnaeus marks the starting point of modern taxonomy. Linnaeus organized flora and fauna as a nested hierarchy of ranks. He introduced a system of binary species names called trivial names for both plants and animals. For each species, he created an epithet that could be used together with a genus name. The trivial names were intended for fieldwork and education and not to replace the earlier phrase names. The phrase names included a description of the species that distinguished it from other known species in the genus. With an expanded knowledge of the global flora and fauna through the 17th and 18th century scientific expeditions, a large number of species were found and named, and thus more terms had to be added to each phrase name. By the time of Linnaeus, the situation was really bad. Linnaeus counted 8,530 species of flowering plants in 1753. The simplicity of Linnaeus's trivial names revolutionized nomenclature, and soon binary nomenclature came to replace phrase names. As an example, this is Plantago media, whose corresponding phrase name translates as plantain with ovate lanceolate leaves becoming softly hairy, a cylindrical head, and smooth stem. Whereas Linnaeus classified for ease of identification, the idea of hierarchical Linnaean taxonomy as translating into a sort of tree-like diagram was formulated toward the end of the 19th century, well before On the Origin of Species was published. But with the publication of Darwin's theory of descent by natural selection, general acceptance that classification should reflect evolutionary history quickly appeared. But for all the positive aspects of the Linnaean system, there were still substantial implementation issues, the principal one being how to resolve nomenclatural conflict. These conflicts take two major forms: synonymy, more than one name available for a taxon, and homonymy, more than one taxon given the same name. Under what conditions should names be changed? Three major criteria were discussed as mechanisms of arbitration. They were suitability, stability, and priority. Suitability is the idea that the name should best reflect the characteristics of an organism. This, of course, resonated with the old phrase names, and even Linnaeus in some early writings endorsed this criterion. But the suitability criterion soon fell out of favor, and discussion of best practices for resolving nomenclatural conflict revolved around stability, where recent, widely used names are preferred over older, forgotten names, and priority, where the oldest name is preferred. In the early 19th century, Candolle articulated, "The goal of nomenclature of natural history is to be universal, common to scientists of all nations." To achieve this, he advocated a system of rules to make taxonomic nomenclature internationally consistent. This included emphasis on the principle of priority. But international taxonomic chaos persisted. For example, the genus name used to apply to the Hercules beetle was Dynastes to English taxonomists, Scarabaeus to the French, and Geotrupus to the Germans. Such inconsistencies throughout the flora and fauna inhibited international scientific communication. Hugh Edwin Strickland attempted to improve the situation, writing, "It would be highly desirable if an authorized body could be constituted to frame a code of laws for naturalists instead of the present anarchical state of things in which everyone does that which is right in his own eyes." Starting with a position similar to Candolle's, including emphasis on the principle of priority, Strickland progressively led the development of an international system of rules of taxonomic nomenclature. But there was resistance from within the international taxonomic community to the emphasis on priority. This was born primarily of frustration with poor quality descriptions that lacked sufficient detail for practical identification. Yet if such descriptions were published first, the taxonomic community was stuck with the name. When can it be said that a species has been described? Even the most fanatical advocate of the law of priority will not pretend that a species has been described concerning which utterly false notices or erroneous or unimportant indications are given, which so completely fail in characterizing the species that no one is able to recognize it. Type specimens, that is, specimens examined by the author in the course of writing the description, can permit identification when the description is inadequate, and most taxonomists today would agree that using type specimens to determine the application of a species name is the rigorous approach. But such specimens cannot always be unambiguously identified. The first truly international codes of nomenclature were established in 1905 for animals and 1906 for plants, including algae and fungi. Each code selected a different work of Linnaeus as a starting point for the principle of priority. The Zoological Code recommended a specific type specimen be designated, with the museum depository and accession number specified. The codes of nomenclature we use today are direct descendants of these early 20th century documents. Early computers in the 1960s had their most visible impact on classification. Tables of characteristics by taxa were used to cluster most similar taxa together. But just as databases rely on unique keys to tie together various tables of data, computer scientists proposed that unique numbers linked to tables of characteristics would be superior to Linnaean names. In a 1961 article on taxonomy in an increasingly computerized world, T.L. Jean made a number of both serious and whimsical predictions. Among them, he predicted the use of numbers as well as names for all taxa, that nomenclatural systems would become completely logical, that names would become less important, priority would become less important and eventually may be discarded, that these changes will irritate most taxonomists, that taxonomists will rise to fight the machines, and that the machines will win. The union of taxonomy with online digital technology is sometimes referred to as cyber taxonomy. But cyber taxonomy is about more than just making knowledge about biodiversity available online. The revolution in online biodiversity knowledge is driven by a system of data standards that allow bits of information to be moved among databases and online resources automatically. I'd like to illustrate this using an historical analogy based on the history of shipping in the 20th century. In the early 1950s, most of the world's great centers of commerce had docks at their heart. Though ships had been plying the seas for thousands of years, using them to move goods was still a hugely complicated project. At the shipper's factory or warehouse, the freight would be loaded piece by piece on a truck or rail car. The truck or train would deliver hundreds or thousands of such items to the waterfront. At journey's end, loading for the next voyage could not begin until every bit of incoming cargo had been removed. By far the biggest expense in this process was shifting the cargo from land transport to ship at the port of departure and moving it back to the train or truck at the other end of the voyage. Today, shipping relies heavily on shipping containers. The complication of loading and unloading vehicles with an ever-unique combination of sizes and shapes is a thing of the past. Shipping containers are standardized so that the same box can be transported using multiple vehicles. This works because of the universally accepted design standards applied to shipping containers. These have placed shipping containers at the core of a highly automated system for moving goods from anywhere to anywhere for minimum cost. So what does any of this have to do with biodiversity? What I'm going to tell you about now, and which you'll participate in the practical lab that follows, is how we can use standardized XML tags to label the semantic content of particular elements within a taxonomic paper. From there, we can move these various elements around the World Wide Web in more or less any way we can imagine. What makes this possible is a system of standard terms that describe all the elements of biodiversity information. That system is known as Darwin Core, a standard developed by the biodiversity informatics community, especially through the auspices of the Taxonomic Databases Working Group, or TADWIG. Darwin Core includes subsets of terms to deal with the place and time that a particular organism was recorded, physical attributes of the specimen and the institution it is archived in, and its place in the taxonomic hierarchy. So Darwin Core is the shipping container of biodiversity information, capable of moving digital biodiversity data around the internet to various websites and databases. Like the advent of the shipping container, Darwin Core introduces efficiencies with the power to revolutionize our relationship to biodiversity data. To illustrate how this works, here is a publication PDF of a taxonomic paper and the XML code that underlies it. This XML tag tells the computer that this text string is the title of the paper. Here is one of the authors. Here is the start of a taxonomic description. And here is that same taxonomic description online at the Encyclopedia of Life, where it has been uploaded through an automated process, and also here at Plazi's treatment bank. The idea that biodiversity knowledge and infrastructure needed to reinvent itself for the digital online world started to be articulated around 2002. One of the most compelling visions for how to reinvent taxonomy for the internet age came from Harvard entomologist Edward O. Wilson. Wilson imagined an online resource with a page dedicated to every species on Earth. But I'll let Wilson tell you this idea in an excerpt from a TED Talk he delivered in 2007.
Sadly, our knowledge of biodiversity is so incomplete that we are at risk of losing a great deal of it before it is even discovered. For example, even in the United States, the 200,000 species known currently actually has be found to be only partial in coverage, is mostly unknown to us in basic biology. Only about 15% of the known species have been studied well enough to evaluate their status. Of the 15% evaluated, 20% are classified as imperiled, that is, in danger of extinction. That's in the United States. We are, in short, flying blind into our environmental future. We urgently need to change this. We need to have the biosphere properly explored so that we can understand and competently manage it. We need to settle down before we wreck the planet, and we need that knowledge. This should be a big science project equivalent to the Human Genome Project. It should be thought of as a biological moonshot with a timetable. So this brings me to my wish for TEDsters and to anyone else around the world who hears this talk. I wish we will work together to help create the key tools that we need to inspire preservation of Earth's biodiversity. And let us call it the Encyclopedia of Life. What is the Encyclopedia of Life? A concept that has already taken hold and is beginning to spread and be looked at seriously. It is an encyclopedia that lives on the internet and is contributed to by thousands of scientists around the world. Amateurs can do it also. It has an indefinitely expandable page for each species. It makes all key information about life on Earth accessible to anyone on demand, anywhere in the world. I've written about this idea before, and I know there are people in this room who have expended significant effort on it in the past. But what excites me is that since I first put forward this particular idea in that form, science has advanced, technology has moved forward. Today, the practicalities of making such an encyclopedia, regardless of the magnitude of the information put into it, are within reach. Indeed, in the past year, a group of influential scientific institutions have begun mobilizing to realize this dream. I wish you would help them. Working together, we can make this real. The encyclopedia will quickly pay for itself in practical applications. It will address transcendent qualities in the human consciousness and sense of human need. It will transform the science of biology in ways of obvious benefit to humanity. And most of all, it can inspire a new generation of biologists to continue the quest that started for me personally 60 years ago, to search for life, to understand it, and finally, above all, to preserve it. That is my wish. Thank you.
This vision was eventually realized as the Encyclopedia of Life. This project is live and ongoing, but funding problems combined with conflicting visions guiding the development of this vast project mean that the current implementation of the Encyclopedia of Life has drifted away somewhat from its original grand idea. But EOL is emerging as an important source for data on functional traits, which will be discussed more fully later in this series. I spoke earlier about Carl Linnaeus, the founder of modern taxonomic research. He is, in large part, responsible for the profusion of taxonomic names that you may find cumbersome to remember or even pronounce. Ultimately, the idea is that each species should have a unique name that links it to everything that is known about it. This includes all the specimens known for each species, where and when they were found, the collections they have been archived in, their characteristics, and publications where observations have been documented. However, taxonomic names can change, much to the frustration of people who have to use taxonomic names. There are basically two kinds of errors that can be made when doing taxonomic research that can lead to name changes. The first is synonymy, when a taxonomist describes a species as new that has already been previously described. The second is homonymy, when a taxonomic description confuses two or more distinct species under the same name. International codes of rules adopted in the early 1900s and periodically updated since provide standards for deciding how to resolve nomenclatural conflict. At the same time, our understanding of how various species are related to each other is constantly improving. The Linnaean system was adopted as a system for managing knowledge about species. Darwin's evolutionary theory was formalized about 100 years later, but fit naturally into the nested hierarchical structure of Linnaean classification, and the notion was soon accepted that classification should reflect our best inference of evolutionary or phylogenetic history. But sometimes advances in phylogenetic knowledge can have consequences for taxonomic nomenclature. That's what happened as the result of a recent study on the famous fruit fly genus Drosophila. Drosophila is a very large genus containing about 1,500 species and is considered to be in need of major revision. The problem arose when it was determined that the iconic genetic model organism, widely known as Drosophila melanogaster, does not belong to the monophyletic group containing the type species of the genus, Drosophila funebris. The scientists involved in this study were not keen to change the name of such a celebrity species, so they petitioned the International Commission on Zoological Nomenclature for a special ruling that would make the name change unnecessary, but that petition was rejected. A similar situation occurred in the late 1980s regarding another model organism, the zebrafinch. Formerly known as Pophilia guttata, it became clear that the zebrafinch belonged to the genus Taeniopygia. In this case, the nomenclatural change was, you might be surprised to hear, adopted almost immediately and uniformly throughout the field. So whenever we use a taxonomic name, we are linking information about one of the more than 1 million species on Earth for which we have a name. This may mean that an individual of that species was observed at a particular time and place, or it may be something about the physical characteristics of that species, or how it interacts with other species in its environment. But if this information is associated with a number of different taxonomic names, all different in spelling but pointing to the same real species in nature, how do we retrieve all the information known for a particular species? For this, we can turn to a taxonomic catalog. For this example, I'm going to use the World Spider Catalog. To briefly take you through the taxonomic history of one species of spider. The valid name is the name for this species as it is in current use. The author and year of the original publication of this species name follows. The identifier is an LSID, or Life Science Identifier, a machine-readable identifier for this species. The treatment citations list all the published taxonomic literature containing content on this species. This species was originally described under the name Eresus fumosus by Carl L. Koch in 1837. Then, in 1865, his son Ludwig Koch described Eresus bubo as a new species. In 1878, Ludwig Koch published a redescription of Eresus bubo. Redescriptions are common in taxonomic research and typically summarize previously known information about a species while adding additional observations. In keeping with standard scholarly practice, the previous description is cited in the redescription. In 1920, Tucker recognized that Eresus fumosus and Eresus bubo were actually descriptions of the same species. So Eresus bubo became a junior synonym of Eresus fumosus. Note that this means that specimens and observations recorded under the name Eresus bubo now apply to the newly expanded concept of Eresus fumosus. In 1967, Leitinen established the new genus Gondonamino and placed this and other species within it. So this is a change in taxonomic nomenclature to accommodate new understanding about how this spider and its relatives are related to each other. Taken together, these taxonomic treatments contain practically everything that is known about this species, including physical characteristics, specimen records, and information on where this species has been found, and how it interacts with other species in its environment. Taxonomic treatments are always headed by a taxonomic name, the identifier that allows us to accumulate knowledge about that species or a higher taxon. Other common sections that may or may not be present include citations to previous literature on this taxon, formal taxonomic descriptions, diagnosis for distinguishing the taxon from all others, notes on biology and ecology, and specimen records. Descriptions of anatomical details may be accompanied by images depicting them. Obviously, spiders are a modest part of the entire tree of life. So what do we do if we are interested in knowledge on something other than spiders? Well, there is also a catalog of life. This umbrella catalog aggregates many smaller catalogs, including the World Spider Catalog, focusing on particular taxonomic groups and other projects. But this means that the catalog of life is much more uneven in its completeness, especially when it comes to literature citations and the different name forms that have been used to indicate particular species in the past. The World Spider Catalog shares content with the Catalog of Life, but when we go to the Catalog of Life page for that same spider, Gondonamino famosa, we find that the synonyms and treatment citations are missing. So let's think of biodiversity knowledge through the lens of this schematic. Through observations of biodiversity in a place, we can assess how many species we encounter and their relative abundance. Each of these species has many specific characteristics. Some of these are useful for distinguishing one species from another, whether we're talking about morphological characteristics that differ from one species to another, or the DNA sequence in a particular part of the genome. Some characteristics influence the role of a species in the ecosystem. These might include size, color, and resource requirements. Such characteristics can be thought of as functional traits. Places have attributes of their own, which are largely, although not entirely, independent of the plant and animal communities present. These include abiotic factors like temperature and precipitation, elevation, soil characteristics, and the relative availability of certain nutrients. Note that some of these kinds of data are pretty straightforward. A species is observed at a specific place and time. Temperature and precipitation for this location are measurable using standard units. DNA sequencing has been routine for decades. But what exactly comprises a functional trait can involve a certain amount of interpretation. How we define and quantify these traits and which traits we choose to focus on can be somewhat subjective. So let's spend a moment to think about the data we have available on which species have been observed where and when. Proceeding from right to left, the most accessible and useful records are available as parsed digital text that can be queried through one or more databases. The Global Biodiversity Information Facility, GBIF, is the largest such database of aggregated species occurrence records. There are a vast number of records printed in taxonomic publications. This is particularly the case with diverse groups like arthropods, but many of these are not parsed and are therefore cumbersome to query or reuse. In other words, we may have some text in a publication that reports collecting information for a particular specimen, but the semantic meaning of that text, the country and specific location it was collected from, the date, the geographic coordinates, etc., is not parsed and available for querying, aggregation, and analytics. Some specimens in international lending institutions like Naturalis have never been included in databases or publications, but exist only as words printed on paper with a specimen. Such records are only available if someone physically examined the specimen and associated data. Finally, there are specimens in private collections which are available to almost no one. The GBIF database currently includes about 1 billion records. Most records are drawn from museum collections like Naturalis or observation networks like eBird. Here we see the concentration and distribution of records for all animals. Here are all the plant records, about 250 million of them. Here are the birds, represented by more than 500 million records. Here is the order Hymenoptera with about 9 million records. This includes the ants, which are among the best of the high diversity groups for digitized species occurrence records. And here are the spiders, the group that I specialize in, with a modest 1.8 million records. When we compare the number of digital records of which species have been found where to the distribution of described species across major taxonomic groups, some clear patterns emerge. For one thing, about half of the records in GBIF are for birds, which only comprise about 10,000 species worldwide, a tiny fraction of total biodiversity. But arthropods, which make up more than half of the world's known species, are represented in GBIF by a comparatively modest number of records. It's also worth pointing out that 1 billion records translates into only about 2 records per square kilometer for all forms of life across the surface of the Earth, and one of those is a bird. And this is a distortion because we have a lot more knowledge about what has been found where on Earth, but this information is not available in digital parsed form that would allow it to be all added together with the existing database records. We have only a vague idea about how many inaccessible records there might be, but we know it's a lot. So there is a body of knowledge that has been registered by scientists that are not available for query, aggregation, and reuse. In an era of global change, this is precisely the kind of knowledge that would be useful for us as we assess impacts and make recommendations for wise policy. This data issue has serious implications for conservation. This is a famous map of the world's biodiversity hotspots, that is, the global priorities for conservation. Hotspots are defined as areas of high species richness and endemism under pressure from habitat destruction. But this map was not made based on all species or even a broad cross-section of biodiversity. It was based on records of vascular plants plus some vertebrate groups. Of course, given the lack of available occurrence data on the world's most diverse taxonomic groups, what else could ecologists do but use the best data available? But this raises an important question. Do the taxonomic groups used to make this map approximate a general pattern of diversity and endemism that fits across all taxonomic groups, including things like arthropods? We don't really know the answer to this question yet, but there are some indications that should raise concern. For example, a study in Australia looked at the total number of species for 4 taxonomic groups, insects, snails, vertebrates, and the plant family Lauraceae, across a reasonably large area of wet tropical rainforest. The study area was divided into 23 subregions, and species present for each of the 4 taxonomic groups in each subregion was determined. These data were then used to assess conservation priorities by ranking the subregions in such a way as to maximize the total number of species. The question can then be asked, does the ranking of subregions based on one taxonomic group predict the rankings for the other taxonomic groups? If so, it might be possible to rely on a subset of taxonomic groups as a proxy for all biodiversity, just as the conservationists have implicitly used vascular plants and vertebrates as a proxy for terrestrial biodiversity conservation priorities globally. What this Australian study found was that cryptic and diverse groups like insects and snails were good predictors of the conservation priorities of vertebrates and fair predictors of the conservation priorities of plants, but that neither plants nor vertebrates did a good job of predicting the patterns in insects and snails. This is presumably because diverse taxonomic groups tend to have finer scale distribution patterns and higher local endemism than vertebrates and plants. So the taxonomic groups that have the greatest data deficit, including diverse arthropods, may be among the most powerful indicators of ecosystem priorities. To assess changes in species distribution in response to climate change, we need to understand where they lived historically to recognize evidence of change. Once we know where a species has been found historically, we can use records of things like temperature and precipitation to develop models of the environmental tolerances of various species. We can apply models of future climate change to extrapolate how species ranges might respond to climate change in the future. And we can use functional traits to predict which species may be more or less sensitive to climate change. So how do we improve the situation? How can we get more of the valuable knowledge that biodiversity scientists have accumulated over generations into a form that will allow it to be combined with the information we already have in the biodiversity information sphere? The taxonomic literature is a rich, high-quality, and largely untapped source of biodiversity knowledge. The gold standard for taxonomic research is the taxonomic revision, typically covering all species from a large geographic region or the entire world. This entails reviewing all specimens from the world's major museum collections for a particular taxonomic group, say a genus or small family, as well as all the previously published taxonomic treatments. Each specimen is associated with data about where and when it was collected. By studying the characteristics of these specimens, all of these specimens are sorted into a number of species that can be distinguished from each other. Each of these species has either been described previously or not, meaning it is new to science. Some of the specimens in all these museum collections were used by earlier authors to describe species. In most cases, particular specimens have been designated by the original authors as the type specimen, that is, the voucher specimen tied to a particular species name published at some time in the past. Some of the species in your global taxonomic revision will have been described previously, possibly several times. The rules of nomenclature help to determine which name should be applied to such species. The rest receive new names. Because these specimens were studied in a comparative context, the species identification of each specimen studied is often the most accurate available. It's easier to get it right when you have specimens representing all of the species in the world of a particular taxon available for direct comparison than it is for a taxonomist using a publication to identify a particular specimen. One solution is semantic enhancement of taxonomic literature. The software package Golden Gate was developed by the Swiss organization Plazi to read taxonomic papers and apply semantic tags to mobilize the unstructured data within. One of the richest and most fine-grained data classes are the specimen records, which we refer to as materials citations. Here we have a taxonomic article marked up in Golden Gate. Some of the data classes that have been marked up for this article are visible on the right side of the screen. Here we have highlighted a few of the fields associated with material citations, such as collecting country, collecting date, and geographic coordinates. Golden Gate allows us to view and edit the values for any of the available fields. Once we're finished marking up an article in Golden Gate, we can upload it to Treatment Bank. Once available on Treatment Bank, the material citations data are available in various forms. If geographic coordinates are available, specimen records can be plotted on a Google map. Also, graphical summaries of the specimen data are plotted. Treatment Bank shares its data with GBIF, so every once in a while a computer program recognizes that there are new material citations data on Treatment Bank, and these data are harvested by GBIF. The IUCN, that's the International Union for Conservation of Nature, produces the Red List, the world's most comprehensive inventory of the global conservation status of species. It uses a set of criteria to evaluate the extinction risk of thousands of species. These include rate of decline, population size, area of geographic distribution, and degree of population and distribution fragmentation. Assessing these criteria are dependent on records documenting where and when a species has been recorded. For many species on Earth, these data are currently available only in the taxonomic literature and cannot be found in any database. Our accumulated biodiversity knowledge includes an estimated 2 to 3 billion specimens in natural history collections and 500 million pages of printed text. These are the data we need to answer questions that are relevant to our world today, like setting conservation priorities and anticipating the effects of climate change on biodiversity and ecosystem functions that affect the lives of people. Computer models of the biosphere are becoming increasingly sophisticated and powerful, and this field appears to have tremendous potential for growth in the near future. To fully realize the benefits of these nascent technological advances, we are going to have to wrestle with the gap between the knowledge we have accumulated in libraries over 250 years of research and the data that are available in structured digital form so they can be used by computers. In short, we have 500 million pages worth of biodiversity knowledge and no way to query it. XML tagging, whether applied to prospective cybertaxonomic publications that prioritize data mobilization or to legacy publications using tools like Golden Gate, makes the primary data available in digital structured form. This enhances the scientific quality of the work because it is easier for other scientists to reevaluate and test conclusions based on the data. But the real power comes when the data from many articles are combined, queried, and reused for new purposes. Potential applications for these data span the scientific, policy, and public spheres. When we all have better access to the information that already exists in the Global Biodiversity Library, this helps us do a better job of exploring what we don't know and wisely applying what we do.